Hi, everyone. Welcome to our virtual NeuroFest event. My name is Kim McAllister, and I'm the director of the Center for Neuroscience and a professor in the departments of neurology and neurobiology, physiology, and behavior. And I want to welcome you to our event. We're so glad that so many of you are here for NeuroFest, even if it's not in person. As many of you know, we had to cancel NeuroFest for the first time last year, right at the start of the pandemic. And oh boy, what a year it's been, right? We hope that all of you and your loved ones have been able to stay safe and healthy during this turbulent time. And we hope to be back in person for NeuroFest next year. So this is one of my favorite events, and I'm really excited to participate in our first virtual NeuroFest. Our truly awesome staff, students, and event co-chairs got creative and put together an innovative 2-day program for you. So we have a lot of fun in store, and I'm going to start by sharing my slides and telling you about what's in store. So what you're going to be experiencing today is day one of our 2-day program. I'm going to start by giving you a sense for what research is at UC Davis and especially at the Center for Neuroscience. And then I'm going to give you a background on the challenges in mental health research. That's going to be followed by 2 short talks by fantastic speakers, Dr. Megan Dennis and Dr. Randy O'Reilly. And then we're going to turn to a student data blitz. It's going to be a real treat. You're going to get to hear from 3 of our awesome grad students. And then we are really excited to share a number of interactive booths with you through the Gather platform. Okay, so we're all here today because we're fascinated with the brain and how the brain makes us human and allows us to do all the things that we can do. Neuroscience is the science of understanding how the brain works. And this is the focus of research at the Center for Neuroscience on campus, where many of us work. Since many of you are new to this event, I thought we'd provide a short introduction to the Center for Neuroscience and neuroscience research in general through a short 4-minute video that I will show now.
We're interested in understanding how the brain works for a number of reasons.
Oops. Ooh. So sorry.
We're interested in understanding how the brain works for a number of reasons. And the first is really based on an innate curiosity that we all have about who we are as humans. Our brain development and our brain function allow us to do all of the amazing things that we do, to learn, to make mistakes, to adapt, to plan for the future, to love, to interact. It really is about who we are as humans.
Figuring out the mysteries of the brain is one of the grand challenges of science. Ultimately, if we're going to solve this grand challenge, we need to bring together people from many disciplines. We need the biologists, we need computer scientists who know how to handle big data. We need statisticians who know how to infer patterns from these data. We need physicists and computational modelers who know how to integrate across all of these different brain areas and across the different scales from molecular to systems to cognitive to put together a holistic picture of how brain function is occurring.
Well, the brain's important because it is one of the most amazing and complex phenomenon in the known universe. We have 100 billion living cells that are kind of organized in these networks, all wired up together, and they're connected to one another through some trillion of connections. And somehow or another, through their coordinated biological activity, we're able to see, I can see you, I can hear you, and I can think my thoughts and have my feelings and communicate with your brains. And that is one of the great miracles we need to try to understand.
The Center for Neuroscience is a very exciting place. It's an interdisciplinary center here at UC Davis that brings together faculty from many different colleges and schools and many different departments. We cover all aspects of neuroscience research, from basic to translational, from molecular to cellular to systems to cognitive neuroscience. It's an exciting place because each faculty member brings together a team of neuroscientists within their lab that have specific backgrounds and perspectives on how to study how the brain works. And when we bring these together from all of these differing backgrounds, we have sparks of new discovery and new ideas that you don't have if you're housed in separate places. We still only know the very tip of the iceberg about how the brain works. Brain disease is incredibly impactful to individuals that have it because many brain diseases prevent us from being able to do what we do as humans, from being able to function in the world the way that we do. And some brain diseases are even more devastating because they start to impact our identities and our ability to interact with our loved ones. So we want to come up with new cures and ways to prevent disease. There are numerous brain diseases that affect almost every family, and we have very few cures or treatments. And here at the Center for Neuroscience, we are trying to understand how the brain works and goes wrong in disease so that we can come up with better treatments and ways of diagnosing brain disease much earlier. So the future of neuroscience is all about team science. It's about innovation. It's about speeding up discovery. It's about taking discoveries and turning it into impact. It's about improving lives.
Center for Neuroscience is the future of neuroscience.
Great. So you can see that the research that we do at UC Davis is really broad. And we typically choose one area to focus on during NeuroFest. And as you all know, this year's topic is mental health, which itself is a very broad topic. Mental health is essential to us, for all of us, to function on a daily basis. And we take it for granted unless and until we experience disruptions to our mental health. These disruptions can come in many forms, including some of those shown here on this slide: ADHD, autism, schizophrenia, anxiety disorders, depression, substance use disorders, and PTSD. And honestly, mental health is at the top of everyone's mind these days. The rates of all mental health disorders have dramatically risen during the isolation and stress brought on by the pandemic. And they were already too high even before COVID. So the statistics that you see here are actually pre-COVID. Psychiatric illnesses affect 15 to 20% of the population worldwide. That includes about 1% that suffers from schizophrenia, almost 7% from major depressive disorder, almost 3% from bipolar, and more than 4% that are affected by ADHD. And neurodevelopmental disorders are also really highly prevalent. Today, autism affects 1 in 54 children and 1 in 34 boys, making it more common than pediatric cancer, diabetes, and AIDS combined. And as if that's not enough, as if it's not enough to have so many people's lives that are directly affected by neurodevelopmental and psychiatric illnesses, there's really a ripple effect of mental illness that's shown here in this slide, taken from the National Alliance on Mental Illness site. So people with mental illness don't just struggle with mental health, but they also have an increased risk for a wide range of chronic diseases like diabetes or cancer. And families are also affected. Caregivers can spend an average of 32 hours per week providing unpaid care. And as if that's not enough, challenges with mental health affects our entire communities. Look at these numbers. 37% of people that are in jail in state and federal prison have a diagnosed mental condition, and 70% of youth in the juvenile justice system have at least one mental health condition. So think, if we could come up with better treatments and even maybe cures for mental illness, the kind of impact that could have on our community and even on our world. Depression and anxiety disorders cost the global economy $1 trillion each year in lost productivity. So despite the prevalence and devastating impact of psychiatric disorders, there remains a pervasive stigma that prevents us from talking about it openly. And more important, it prevents people from getting the help that they need. It can take years for people to seek treatment, if they ever seek treatment at all. This needs to change. We need to stand up against stigma. There can be no health without mental health. And why is it that there is such stigma around mental health disorders when there's little stigma to other diseases? Why do we approach our mental health differently than the health of our bodies? It would be silly to think that we could improve heart disease by telling ourselves that it's not really a disease and that we should just be stronger and power through whatever is happening within our heart. So why do we do that when we experience issues with our mental health? It's important to realize that mental health is brain health. So we have to better understand brain health in order to develop new treatments to improve it. And just like for heart disease, brain disorders are caused in large part by things that we cannot control, like genetic mutations. But for brain disease, it's different genes that are involved. And you're going to hear more about how changes in gene expression during brain development can cause autism spectrum disorder in the first talk that follows my introduction by Dr. Dennis. So it turns out that many psychiatric illnesses actually start early in development, even though the symptoms appear later. And those changes in genes that cause disease actually converge on changes in the number of connections or synapses that are made in the brain. And so what you'll see here is synapse number over time, and it actually changes over time. So if we plot the number of synapses that we see over time, what we find is that after birth, there's a really rapid increase in the number of synapses during infancy and childhood that peaks around age 9 in humans. After that, these connections are refined. Some synapses are strengthened and others are eliminated during adolescence and probably into the late 20s, after which synapse number remains relatively stable. So what happens in disease? In schizophrenia, for example, one of the most enduring and reproducible findings is a decrease in the number of synapses across multiple brain regions. And it's thought that for autism, the change in synapse number starts a lot earlier, and there's an overgrowth in the number of synaptic connections. So those changes in synapse numbers also come with changes in the information that's transferred across them. That information transfer is mediated by chemicals called neurotransmitters that convey the information. So when information starts to come down one cell, that leads to the release of these little spheres that contain the chemicals called neurotransmitters. Those neurotransmitters bind to receptors on the other side of the cell and therefore transmit information. And you probably know the names of some of these neurochemicals, like serotonin and dopamine. And we do have drugs that target these neurotransmitters, like SSRIs that are used for depression. And those drugs can dramatically improve people's lives. But they don't improve lives for everyone. There are many people that do not respond to any currently available drugs. And so as researchers, we're still trying to understand how to better fix the imbalances in these neurotransmitters and how to target treatments specifically to specialized circuits that mediate the behavioral effects that we know as mental illness. So the second talk today by Dr. O'Reilly will illustrate an innovative way to use computational modeling to make predictions about which circuits are involved in diseases like schizophrenia, and even to screen for the efficacy of specific therapeutics. So a shared goal for many of us at UC Davis is to develop more effective diagnostic tools and treatments to improve the lives of so many people that are suffering from changes to their mental health. We can't achieve that goal without discovery-driven neuroscience research. And the way that this research is central to developing new treatments is illustrated on this slide. The way that this works is that we characterize our human disease, and then we identify what causes it, maybe a genetic mutation. And then we create a disease model, maybe in an animal or maybe in a computational model that actually mimics the effects of that disease cause. And then we study through basic science what goes wrong in the disease. And what goes wrong can tell us about what new targets we might want to focus on to develop new drugs to rescue the things that are going wrong in the developing brain. So we test those novel therapeutics in our disease models, and eventually we get to testing them in humans, and that's how new treatments are developed. Okay, so With that, it is time for me to introduce our next speaker, our first speaker, actually. So for these talks, I know that many of you will end up with questions. We would like to point out that it would be great if you could use the Q&A function on Zoom to ask those questions. We can monitor the chat for technical issues, but Q&A is great for questions. Okay, so our first speaker is Dr. Megan Dennis. She's an assistant professor in the Department of Biochemistry and Molecular Medicine in the UC Davis School of Medicine. Through her joint affiliations with the UC Davis Mind Institute and the Genome Center, she and her team seek to identify genomic contributors to human neurologic traits and diseases. Specifically, her lab uses a variety of techniques, including genome sequencing, bioinformatics, comparative genomics, cell culture models, and zebrafish to characterize the functions of genes and mutations. The title of her talk is Fishing for Neural Health and Disease in the Evolutionary Gene Pool. Megan.
Sorry, guys, I'm muted. Okay, thank you so much for joining us for NeuroFest. So today I'm going to talk about some of the work we have ongoing at UC Davis, in which we're using zebrafish to model genes that we identify from sequencing screens that are important for neural health and also for disease and also evolution. And so when we talk about genes, the first thing we need to do is be able to decode our genome. And so our genome is encoded within DNA. So when we look into every single one of our cells, we have DNA within our cells. When in my cells, I have the same exact DNA in every single cell in my body. Every single cell in my body. So I can, what we can do is we can extract some kind of cell from our body. So typically what you would do is you could extract cells from your mouth, and so you can spit into a sample and be able to get cells from your mouth, or you can extract blood. Then what you can do is you can burst those cells open and extract out the molecules that are DNA. So then what we do is we have these really long microscopic molecules, and we'll need to fragment them to be able to decode them. We take these molecules and we stick them onto a sequencer. And so the sequencer that we typically use is called Illumina, and it's Illumina sequencing, and it generates billions and billions of pieces of information that's decoding the DNA that's within our cells. And so what it ends up looking like is that our DNA are encoded as 4 different letters we call nucleotides. And so we just get billions of these different DNA sequences that represent our genome. Human genomes comprise 3 billion nucleotides. So when we compare across different humans, we actually see that humans, even though we look very, very different, our genomes are very, very similar. So we're 99.9% similar amongst all humans. And so we would expect to find a difference between you and a complete stranger once every 1,000 bases within our genome. But what we can do is, now that we've sequenced our genomes, we can look for the differences that exist. And the differences that exist, which we call variants, those are the things that contribute to the differences that we see between you and I. And so here's an example where we have this person 1. We can go across the genome and we can find these different nucleotides that are represented as letters. We can look at person 2 and look at their genome, and we can line them up with person 1. And we can find that for the most part, we're the same, but there are some sites where we have variants. So we have here a variant here A, variant here C, and those could be contributing to the differences we see between these 2. Person 3 also has differences, and in this case, person 2 and person 3 have a similar difference, and that similarity could actually represent similarities that we see between the 2 of them. And then we can look at this person 4 and look at that. Person 4 also has the same variant at the site. And if we look at person 2, 3, and 4, we might say that the difference in hair color, this purple or this orange hair color, might be associated with this particular variant. And that variant could be contributing to the hair color that we see between these 3 that we don't see in person 1. So this is a very simple way to describe how we actually use the DNA sequences to try to connect the variants we identify with traits that we see in humans. So we've done this sequencing across across just hundreds of thousands of children who have autism and epilepsy to be able to do a very similar analysis where we look for variants across the genome and try to find variants that are found in children who have autism or epilepsy that might be contributing to their neurodevelopmental differences. And so doing this, we've identified thousands of variants and we've identified them in hundreds of genes. And so there's just so much information there. Many of these genes we know are important in brain development. So it's very exciting. We can start to try to understand the underlying mechanisms contributing to the differences we see in these children. But the thing is, even though we don't have many differences between us, we have 3 billion nucleotides and we have one variant every 1,000 bases. So there's lots and lots of variants to search through. And not all variants that we find actually cause disease. And so what we're really interested in understanding is how we can actually test if a variant that we identify from a sequencing screen actually leads to dysfunctions of the brain that then leads to differences in these children that have autism or epilepsy. And so the way that our lab does this is by using zebrafish as a model organism. And that might seem very strange because we're looking at these human-specific traits of neurodevelopment and cognition, and how is it that a zebrafish can actually model this? And it is a little complicated because, how is it that we can do this? But the reason we can do this is because humans and fish actually share a fair number of their genes in their genomes. So humans have on the order of 20,000 genes in their genome, and zebrafish have 70% of those same genes in their genome. And that's because of evolution. So about 400 million years ago, we shared a common ancestor with fish. So our great-great-great-great-great-great-great-grandparents were the same. And so because of that, we have a lot of the same genes. And so if we have the same genes, we can attempt to try to humanize fish by introducing variants we identify from our sequencing screens into fish and observe their impacts on brain development and on behavior. But why is it we use zebrafish? So we could use any kind of animal. A lot of folks are using mice, but in our lab, we use zebrafish. And you might recognize the zebrafish here. You can find them at any old pet store. And we actually have some zebrafish in our house at home. So why is it that we use them? And so some of the reasons we use them is because zebrafish can produce many babies, and those babies can be very easily collected. So they produce eggs, and between a mom and a dad in a single fertilization event, they can produce 200 babies. And so if you have 10 crosses ongoing, you can get 2,000 babies in one morning. And so that allows us to characterize many, many genes at the same time and do lots of different experiments. And further, these babies, they grow extremely quickly. So this is a time-lapse video showing the first 18 hours of development. So in less than a day, we have this single cell organism that started as an egg that has this, by the end of the day, has this yolk sac, has a spinal cord, has a tail. And by the end of that day, we have a fish who has an eye, a heart that's pumping, the beginning of a brain, and we have these somites here. And by 5 days, our fish are looking like little fish. They're eating, they're swimming, they have little brains that we can test for neurodevelopmental defects in them. And so we can do our experiments very quickly. Additionally, we can house thousands of fish in a really small space. So this is a picture of our laboratory over on the UC Davis campus, and it's a really small space. But we can house, this little facility can house up to 7,000 fish. So we have 25 fish in each of these little containers, and combined we can do lots and lots of experiments. And so if we know that a single cross can make 200 fish, that makes a lot of babies. So the way that we go about incorporating these human variants into fish is by using this approach called CRISPR. And so we take this CRISPR, which will allow us to specifically target any gene in the zebrafish genome. We inject it at the single cell stage. So we collect our fish, they're external, the fertilization, so we can grab the eggs immediately. This is what a single cell embryo looks like. It's got this chorion, or its shell. We have a zygote here with a single cell, and then we have the yolk. And if this is a transparent egg, it's really easy to actually pierce the yolk. And so we take a little micro needle and we just inject CRISPR directly into the egg. And that CRISPR will target the gene in the DNA and alter it to a state in which we have a human variant in that fish. And so the next step, once we create these mutants, we'd like to be able to test for changes in the zebrafish brains that may mimic what we might see, what we might expect to see in a child with autism spectrum disorder or with epilepsy. And so we have a number of different tests that we use in our lab to be able to get at that. So we can test, for instance, in the case of epilepsy, for seizures in our fish by tracking their motion. And so we do this by using this little apparatus here called Vision. We can place a 96 well plate onto there with a single 5-day-old fish in every single well, and then we can treat them with a drug that then induces epilepsy in these fish. And the way that looks, how seizures look in a fish, is that there's different stages we can detect. So in this case, we have the stage 1 seizures, and what we're showing you here, these little dark dots are a top view of the fish moving around in each of their little circular wells, the dark part of their eyes, and then you can see the tails behind them. And so when a fish is having a seizure, what it looks like is that they suddenly have this hyperactive movements and just kind of this burst of movements. And so that's what's shown here for stage 1 seizures. The next stage is stage 2, where a fish really has a lot more activity and is way more hyperactive and starts whirlpooling around its well. And then the most severe stage is stage 3, where the fish are actually moving less and there's a loss of posture and they have periodic spasms. So for stage 1 and stage 2, a seizure would look like increased movement. And with stage 3, a seizure would look like reduced movement. And so we went ahead and we tested variants of a gene called SYNGAP1, which is a gene that has been mutated in children with autism and also with epilepsy. And the mutation creates a form of the gene that no longer works. And this gene is important in the connectivity and the communication between neurons. And so when we take our fish that haven't been mutated and we treat them with this drug, PTZ, we see that the fish are moving and they're hyperactive. So you can see this is distance moved and this is the time that we're testing them. But when we also do the same exact treatment with our mutant fish, where we introduce the SYNGAP mutation into the fish, we see these fish move a lot more. So they're way more hyperactive. We're seeing way more whirlpooling going on. So that's more of that stage 2 seizures that I was describing before. And so this is showing us that the mutation that we identified in SYNGAP1 in patients, when we introduce that into fish, it also leads to epilepsy in these fish that the patients also exhibit. We can also do things like measure body features using imaging. And so again, this is a really nice feature of zebrafish because they're so little, we can do this in a really high throughput way. So what we do is we use this cool system called the VAST bioimager, where we have a 96 well plate of these 5-day-old fish. We suck them up into a little tube. We automatically put them under a microscope and we orient them into all these different orientations. And then subsequently, we can extract various features automatically from these fish, such as area of the eye, width of the head, length of the fish, overall area of the fish, to be able to determine if there's any kind of changes in their growth or changes in the size of their eyes or their brains. We did this for SYNGAP1, and what we found was that in that same exact mutant, we found that there were differences in the size of their brain, and they had arrested development, so they weren't growing as quickly as the other fish that didn't have the mutation. Another area of interest in our lab is not only looking for genes that are important in disease, such as autism spectrum disorder, but we're also really interested in identifying genes that are unique to humans that have led to changes in our brains and behavior that are unique to our species. And so when we look at humans and look at our most closely related species siblings, such as chimpanzees or gorillas, we can see that there has been a drastic change in the sizes of our brain in the last 2 million years in the Homo sapiens species, in the lineage. We also know that the neural networks and the connections between neurons are much more complex. And so we'd like to know what the genes are that are underlying that. Because of these differences in the size of our brain and also the differences in the connectivity between neurons, that's allowed humans to have these really remarkable features, such as the ability to read and have complex language. So we wanted to go ahead and use our zebrafish to test a gene called SYRGAP2 that has been found from genetic screens by comparing human sequences to genome sequences of gorillas and chimpanzees. And we found variants that were unique in humans. We know this gene is likely important in brain development, and we think it might be important in the expansion of the brain. So we went ahead and made a human version in a fish. When we did this and we put that fish through that VAST bioimager, what we were able to show is that our fish that were typical without the mutation had smaller head sizes compared to the fish that contain the human-specific variant. So when we introduce the human-specific variant, we see fish who have larger heads, and that's likely because they have larger brains. We went ahead and we also characterized the number of neurons within the brains of our fish. And this is again at 5-day-old fish. And this is a top view of their head looking down. We're looking at this as a midbrain, forebrain, and hindbrain. The little green dots are representing neurons within this fish. So this is a typical fish, and we can count the number of neurons that are existing within that typical fish. And we can compare it to our SIRGAP2 mutant that's humanized, and we can see that there's far more neurons that are being exhibited in this fish. And we can quantify that, that's shown here. And that's really what we're seeing in this increased head size, is that we have way more neurons that are also appearing in these fish. And so overall, we think that zebrafish are a really exciting and excellent model to be able to characterize genes and variants that we identify from sequencing screens. So we're using it to identify genes important in disease and also genes that make us uniquely human. And the ultimate purpose of identifying these genes and these variants is to be able to identify children at a much earlier age that may have autism spectrum disorder or epilepsy or other neurodevelopmental disorders. So the dream would be to have a child and sequence their genome very early on and be able to identify these variants within genes and feel confident that a variant within that gene could lead to some kind of neurodevelopmental disorder that then we can intervene earlier with various different treatments. Also, zebrafish are an excellent model to test for possible treatments in the form of drugs. And so because fish live in water, if we find a fish that is exhibiting epilepsy, for instance, what we can do is we can put all kinds of different possible drugs within the water and see if any of the drug treatments can rescue that epilepsy trait. And we can do this in a high throughput manner. And so that concludes my presentation. And this is just a picture of my lab in December and a recent lab meeting that we have since we're living in the age of COVID. And so everything is done during Zoom. And I'm happy to take any questions you might have. Thank you.
So Dr. Dennis, that was an awesome talk. Thank you so much. We have a couple of questions in the Q&A. If anybody has questions, please add it to the Q&A and not the chat so that our speakers can answer that live. But we've got a couple of minutes for questions. So the first one we'll take from the pre-submitted questions that people submitted during registration. And the question is it possible for genetic mental health disorders to be fully cured?
Right, that's a great question. So I think the core component to that question is that they're genetic disorders. So there's something that exists within the DNA. So to be able to cure something, a genetic disorder that is a change in the DNA, you'd have to be able to change that site in the DNA. So I would say that genetic disorders can't at this time fully be cured. But there are certainly many things we can do to help alleviate genetic disorders. And so, we can, like I talked about before, we can find drugs that can help treat various different features of the disorder, such as if you have epilepsy, you can have certain kinds of drugs that can help with seizures. Also with autism spectrum disorder, children also commonly have gastrointestinal disorders and things like that, sleeping disorders. And so we can try to really target those kind of things. But I would say that, especially in the realm of neurodevelopmental disorders, being able to fully cure them, quote unquote, cure them, is not something that we can do. But certainly we can try to diagnose them earlier, intervene earlier, and try to help alleviate symptoms.
And your work is really exciting in making progress toward those goals. So thank you. We have a question in the Q&A from Vanitha. The question is your work focused on WES and RNA-seq, which is expressed genes, or does it include SNPs in introns and non-coding regions?
Yeah, that's a good question. So I talked very broadly about sequencing the genome. And so there are different ways of being able to sequence the genome. And so the genome is 3 billion bases, but most of it is non-coding. And then there's a small number of regions that are encoding these genes that then create protein. And so for the most part, we focus on the genes. And so that's this whole exome sequencing, or WES, is what you're talking about there. So it's sequencing specifically the genes within the genome. And then the RNA-seq is actually understanding how much of a gene is being transcribed and if there's more or less of it being transcribed. And so that's something we might also look at as well and compare across different individuals. So mostly we're looking at the genes themselves. And we look for variants within the genes. And so we do look for SNPs, I think is what you have there. So these SNPs is another term for a variant. And so, yeah, that's it. But then, there's really interesting research ongoing at UC Davis where folks are looking also within non-coding regions. So I'm looking for protein coding changes, but then there's also the kind of variants you can identify that could change the amount of a protein being expressed. And so that can be from variants in the space between the genes. And that's the quote unquote non-coding space.
Okay, terrific. Another question that's popping up in Q&A from Lisa DeSanti. Can you also examine in your lab the effect of environment on gene expression in regard to mental illness?
Absolutely, yes, especially with the zebrafish model system. And I think that's something for us that could be interesting next steps is, oftentimes, it's not these disorders, it's not just a genetic perturbation that is contributing to these differences, but it also could be the combination of genetic and environmental components. And so that's something that we can do. We can create fish that carry these variants that we identify in children, and then maybe we add some kind of chemical that maybe the child could, that children could have been exposed to at an early age, or air pollutant of some sort into the water, and see if we have more extreme differences or more extreme changes in the neurodevelopmental features of the fish. And I think that's a really strong, that's a very powerful technique to use with the fish. I think the fish are great for looking at environmental factors as well. Yeah.
Yep, and I just would add to that there is a lot of research at UC Davis that's focused on environmental influences and neural immune influences on brain development. So this is a great place if you're interested in that. Okay, we've got one last question for you, Megan, I think. This is from Deborah Neff. Hi, Deb. How long does it typically take to test a mutation of interest, introduce it into the zebrafish, and look for effects you can describe? And how often do you need to reproduce the experiment to determine it is a true cause and effect?
Yeah, so that's very specific, and I can get into the details a little bit here. So the amazing thing about zebrafish is that what we can do is we can introduce a CRISPR at a single cell stage and report defects 5 days later. And because we have so many fish that can be produced at once, we can do this highly parallelized. So we can test hundreds and hundreds of fish in a single injection day. If I have a really motivated student at the time who's ready to inject that many fish. And so, we can start to get traits within 5 days where we can start to feel we're showing significant association. We typically like to test across multiple different crosses. And so we would like to repeat an experiment just to ensure that we were able to recapitulate what we found. And so I would say, we could get results within kind of 7 daare 8 days, a week, 2 weeks. We can start to feel like maybe this gene is doing something.
Okay, fantastic. So there are several more questions, Dr. Dennis. So if you could stay on and type in the answers, whatever you can't get to, we'll follow up with. Thanks, everyone, and thank you so much. Dr. Dennis, you need to look at some of the comments about your talk in the chat. They're really fantastic. So thank you.
Awesome. Thank you so much, everyone. I really enjoyed this.
Okay, great. So now we're going to turn to our second speaker. Dr. Randy O'Reilly is a professor of psychology, computer science, and neuroscience. He joined the UC Davis Center for Neuroscience in 2019. Dr. O'Reilly is internationally recognized as a founder of the field of computational cognitive neuroscience, and he develops large-scale systems neuroscience computational models to learn how neurons give rise to human cognitive function and to inform our understanding of brain-based disorders such as schizophrenia and Parkinson's disease. The title of his talk is Dopamine, Schizophrenia, and Decision-Making. Dr. O'Reilly.
Great, thank you so much, Kim. This is really great to be here to talk with you guys about this. So I'm going to be talking about schizophrenia, and a lot of people are familiar with schizophrenia from its more kind of florid, vivid aspect of hallucinations. And when you think of schizophrenia, you think about these kind of what we call the positive symptoms of schizophrenia. But in fact, people who suffer from this disorder really are more impacted by what are called the negative symptoms. And these are characterized by a list of these kind of symptoms: apathy, withdrawal, inability to experience pleasure, impaired attention, etc. And these are actually some of the same effects that you see in major depressive disorder. And what we're trying to do in my research is try to understand how these kind of motivational factors arise from the brain. And one thing that we know across a lot of these disorders is that there are these kind of shared symptoms. And so if we can understand what's happening with the negative symptoms in schizophrenia, it also potentially gives us some insight into what's going on in major depressive disorder and other cases where you see these same kind of symptoms. And so what I'm going to do is give you a little bit of a overview of how we use computational models, which is kind of at the very high level from what Megan was talking about, trying to integrate multiple brain areas into an overall understanding of what's going on across the large scale circuits of the brain. And in particular, look at the circuits that are important for decision making. And we have a little example from Homer here, and then use that to understand what's happening in schizophrenia and other disorders. So the key idea about how you make a decision is actually fairly intuitive. And you can kind of look at this and say, wow, I don't know if I needed a model to tell me this, but in fact, we do have a really thorough understanding of how this works in the brain. And it's very much like this kind of classic cartoon that you have these two competing kind of forces that you're weighing. Okay, you're deciding, should I do this thing or should I not do this thing? And you're kind of weighing the evidence against those two decisions. And of course, Homer always picks the wrong choice, but anyway. And the critical question that we've been focusing on is how learning works in this context. And you've probably heard about the neuromodulator dopamine. It gets a lot of press these days, people talking about going on a dopamine vacation and all this other kind of stuff. And the really interesting thing about dopamine is that it drives learning in your decision-making pathways. And so a lot of our computational models have been built up to understand how this works. And it's actually a really intuitive principle, okay? So if you make a decision that turns out good, better than you expected even, okay, that's when you get a burst of this dopamine. And the dopamine comes up and reinforces the brain systems that were important for making that positive decision, right? So do something good, experience dopamine, and then it kind of reinforces that decision. Oh yeah, that was a good decision. And of course, you have to have the converse. If you make a decision and it turns out that something bad happens, worse than you expected, this dopamine coming from that kind of negative feedback will reinforce the opposite factor in that decision-making process. And so next time you're facing a similar kind of decision, you're going to look at that and say, gee, last time that didn't turn out so well, maybe I shouldn't do that. And of course, if that's only if you're not Homer. And so this kind of learning process of having a signal that is measuring how well you're doing in terms of dopamine, and then using that to drive these kind of two different pathways to make our decisions is really what we understand is going on in the brain. And so this is a diagram of the basal ganglia inside your brain here. So, situated inside of Homer's brain. And here are these two different pathways. They're the go pathway and the no-go pathway. And they're affected by this dopamine coming from the brainstem. And the dopamine affects these two different receptors, the D1 and the D2 receptor. And it's incredibly complicated to work through. There's all these kind of double negatives, and two minuses make a positive. And I won't torture you with all the details of how this kind of circuit works. And it's part of the reason why we need computational models to understand the complexities of all these dynamics taking place in the brain. But these kind of two opposing pathways really are there. And they're active in every decision that you make. Okay, so and a lot of people talk about this in the context of like the gut feeling. And so your cortex up here at the top of your brain is where you're thinking about all the stuff that you're consciously aware of and all the kind of factors that you're considering in making a decision. And then the basal ganglia is this kind of lower, more evolutionarily ancient system tied in with this dopamine that gives you that kind of gut feeling, like, is this the right thing to do or not? And often people have that intuition that, gee, I should have just gone with my original gut feeling about this, and you know, I was overthinking it from these kind of cortical thought patterns up here, and maybe this kind of wisdom of the accumulated decision-making feedback that you've gotten through this dopamine-based learning process, that kind of is a certain kind of wisdom that gives rise to that gut feeling. And so we use computational models to understand how this circuit works, and we can understand it, you know, from a machine learning perspective. There's a whole field of reinforcement learning that allows us to understand how these things work computationally. But here we're applying them to understand schizophrenia. And schizophrenia is associated with changes in these basal ganglia pathways and in the dopamine system. This has been known for a long time. This is more recent data from Cam Carter's group. And seeing these differences, we can understand that there really are changes in these fundamental decision-making circuits. And our job with the computational models is try to understand how these changes are affecting behavior. And so to understand behavior, you need an assay, like we're talking about in what Megan was talking about with her, you know, video recording of fish. And here we have people, and we can actually do fun tasks. And so if we're talking about measuring motivation, one of the tasks we look at is something that requires a certain amount of engagement of mental effort. And the task is kind of like these things that you may be familiar with from the old kind of like comics in the newspaper, where you have these two panels and you're trying to look for differences. And so, you know, there's 6 differences, and you can kind of stare at this. And if I leave this up, you guys will just sit there and, you know, obsess about finding all those differences. They're pretty subtle. The actual task is a little bit more boring. So we have like a display that shows up, and then one of these different colored blocks will change color. Critically, we kind of blank the screen, so you can't kind of like visually see the difference right away. You have to sort of remember it and then spot this difference. This one changed, and you have to identify which of these colors changed. And what you see is that there are differences in kind of overall capacity to encode how much, you know, how many different squares are on the screen, but also critically a big difference in the effect of attention. So people with schizophrenia show a reduced engagement and an increased kind of tendency to lapse and mind wander and not pay attention to what's going on in this task. And so that reflects, we think, these kind of motivational factors, and they're actually associated with these kind of motivational factors that we measure in the basal ganglia. And so really our computer models provide this ability to link across all these different levels of analysis. So, you know, things that are happening at the level of these neurotransmitters, dopamine, the detailed biological factors, the ways that drugs affect this system, all of those kinds of things can be incorporated into these more detailed computational models. Again, we have the brain circuitry. We can run these models. So the next slide here shows you the model actually runs as a kind of, you know, autonomous little system there. It's getting dopamine, it's making decisions, it's deciding whether to engage in this particular task. And the model literally produces the behavior that we see in patients and healthy controls. And we can use that whole framework to understand the connections between all this neurobiological data, behavior, and critically, you know, pathology in disease conditions. So it really allows us to kind of integrate all of the different kinds of levels of analysis that go on in the Center for Neuroscience and try to put it together in a more systematic way. And I'm always amazed at the amount of work that it takes to you know, figure out all the details of the neuroscience. And I get this kind of enviable position just to sort of sit back and put it into these computer models. But yeah, it really is a pretty amazing process. So thank you for your attention. And these are the folks in my lab and collaborators working on this particular work and funding. Thank you. So I'm happy to take questions.
Okay, thank you so much, Dr. O'Reilly, for a fantastic talk. And for those in the audience, it is not simple to create the models that he just talked about. So he's being quite humble. Okay, so like with the last speaker, we are going to go ahead and take a question from the pre-submitted questions. So we're going to start with Is the pathway for pleasure and anxiety in the brain the same?
Yes, and that's really remarkable. We actually just published a whole paper on this topic with a very detailed model. You may have heard of a brain area called the amygdala, and that's part of this same whole circuitry that the basal ganglia is kind of involved in. And the amygdala originally was thought to be kind of just for negative emotions and kind of aversive states. But more recently, people are really appreciating that it has a lot of pleasure states in it as well. And so it really does seem to be an integrated system. And there's a pretty interesting proportion of cells in the amygdala. About 80% of them are associated with negative outcomes, and 20% are associated with positive outcomes. You can think of this as the kind of Dostoevsky rule that, there's a lot of different ways to experience negative outcomes and not too much variety in positive outcomes. And that seems to be true in general in the brain, that the brain is definitely more concerned with negative outcomes. And of course, we know from mental disorders that most of the mental disorders are associated with increased, anxiety, depression, all these kind of more negative mental states. But in fact, the brain systems really are integrated, and understanding those connections between how the positive and negative pathways kind of relate, it actually connects directly with that kind of go, no-go process. Those are kind of these opponent processes opposing each other. And it turns out that positive and negative outcomes and emotions also have that same kind of, push-pull kind of relationship, that competitive relationship with each other in the brain. So it's actually important for them to be localized in the same brain area.
Okay, terrific. We're going to take a question from Lynn Weaver. The question is, what are the side effects of taking dopamine?
Yeah, so, well, you may have heard, that dopamine is affected by lots of drugs of abuse. So cocaine, most famously, heroin, a lot of substances affect your dopamine regulation. You can't actually take dopamine directly, as far as I know, but yeah, these other substances do affect it. And one of the interesting things about dopamine is that it is a signal of a difference from your expectation. And a lot of people think of it as just like raw pleasure, but in fact, it's actually telling us how different was something compared to what I expected it to be. And the nice thing about that is that if you're not doing anything kind of external to the system, it's kind of self-regulating. So as you start to get better at something, more successful at making good decisions, for example, you learn to expect the dopamine and therefore not get as much. And therefore, that's kind of driving this whole cycle of ambition and greed also. So it has a negative consequence. But that kind of self-regulating aspect of dopamine is really important from a learning perspective. And so when you have a drug of abuse like cocaine or heroin or something like that, it actually short circuits that kind of natural feedback mechanism. And that's a major problem. And that's where you see kind of addiction and dependence coming in, is that it's short circuiting the natural kind of negative feedback loop built into the brain. So that's obviously, the side effect of taking drugs of abuse like that is dependence. You need more and more, you get tolerant. There is some adaptation, some learning that's still taking place. And so you need more and more of the substance to give you the same subjective feeling, et cetera. So yeah, I think those are, and we can understand exactly those kinds of phenomena using the computational models I just described. So that's one of the major areas where people are looking at these kinds of effects.
Okay, terrific. We have another question in the Q&A from Vanitha Dubridge. The question is, do your models predict temporal changes in the ability to focus, for example? So in times of stress, do the profiles change?
Yeah, absolutely. And that's one of the key things that we're looking at. There's really interesting data showing just naturally, according to these longer time frames, a natural kind of ebb and flow of attentional focus. And there's a lot of research looking at this, where people basically, tune in and tune out. You find this subjectively when you're reading or whatever. you experience that kind of ebb and flow of engagement. And then on top of that, the systems like dopamine that we're talking about in the basal ganglia interact with all these other neuromodulatory systems in your brain. So one of the main ones is serotonin, and serotonin and dopamine interact directly. And serotonin, among all the various things that were known to be affected by serotonin, is arousal and stress. And so when you're experiencing stress or you're experiencing different states of arousal, those are all kind of entering into and affecting those pathways in your brain. And so really, again, I find that you need this kind of computational approach to really kind of make sense of and interconnect all those different effects of these different modulators on the different circuit pathways in the brain. That's one of the things that we're really interested in looking at, is how all those different kind of regulation pathways affect each other. Yeah, so very much all of these systems are interacting. And circadian rhythms in particular, one thing, a very interesting line of research shows that, as people age, there's a tendency for a difficulty in engaging the same level of attention and motivation later in the day. If you test more elderly people early in the morning, they actually perform much more similar to younger people. And there's this confound that grad students like to run their experiments in the afternoon because, of course, they're young and have lots of dopamine later in the day. And so if you do actually get people to run the experiments early in the day, you actually see much fewer differences as a function of age, which is pretty interesting.
So I can totally attest to that personally. That is absolutely true. Okay, so we've got more time for questions. Another pre-submitted question was, what's the progress on treating Huntington's disease?
Yeah, so Huntington's disease affects this same system, the basal ganglia, and it's directly that kind of impulsive behavior that you see is driven by that same kind of go no go pathway. It's a genetically driven disease, and as Megan was saying, that it's very difficult to fix those without changing the kind of underlying genetic factor. And so currently, the current research that I saw did not. There's no like approved standard treatment that is effective in treating it. But there are new research strategies looking at kind of using immune-like antisense systems that come in and try to counteract the accumulation of the kind of proteins that build up in the course of that disease. And so there are some pretty interesting cutting-edge treatments that are coming out from understanding directly how these the process has gone awry and really being able to target the treatment directly at it, and a whole new class of these more targeted kind of immune-like systems that can go in and really attack a particular type of protein buildup in the brain. And you may have heard about that in other contexts as well. So that's really the cutting edge kind of treatment right now.
Okay, so I think we've got time maybe for one, maybe two questions. Let's see. So Deb Neff asks, how do you try to capture and identify the various external factors that might impact what you're studying? And how do you spot some factor you didn't expect that's playing a role somehow in your models?
Right. And yeah, so one of the things that I think when we talk about motivation and decision making and these kinds of high level constructs is the really important role of environment and culture and social interaction and all these other factors that come in and shape kind of how we feel about what we're doing, what kind of goals we have that we're trying to achieve through these kind of decision making processes. And so this is something I think is incredibly important, and it really gives us a way to then think about treatments at the level of kind of social level interactions. Therapy is actually remarkably effective because it's a social interaction between the therapist and the client, and it plugs into these kind of natural social pathways we have for controlling our overall mental states. And so understanding that kind of social influence on decision making and how that kind of enters into these brain pathways, right? How do you get some external person out there in the world coming in and affecting how you're making your decisions inside your brain is one of the most important questions we're trying to look at. Yeah, so that's definitely a key aspect of what we're looking at.
Okay, that's terrific. Thank you so much, Dr. O'Reilly. I know that there's some questions both in the chat and in the Q&A that we didn't get to. I'm sorry about that, but if you could stick around and maybe answer some of those, it'd be great.
Yep.
And if you don't get your answers to the attendees, please follow up with us and we'll try to follow up with you if there are questions that you've stuck into the Q&A. So thank you so much for your talk. And I'd like to thank both of our speakers again for two really, really fantastic talks. Now, I want to turn to the next section, which is the data blitz. But before I do that, I want to thank our two NeuroFest co-chairs, Dr. Tim Hanks and Dr. Kostas Zarbalis, who've really done a wonderful job together with Jennifer Scott and our amazing students to organize this event. It's no small feat to take NeuroFest that's huge and turn it into a virtual event. And so despite a few technical glitches, which I hope you're all patient with us about, it's going great. Dr. Zarbalis will lead the next section of our event. That's the data blitz. So he is an associate professor in the Department of Pathology and Laboratory Medicine at the UC Davis Medical Center. Dr. Zarbalis is also a member of the Institute for Pediatric Regenerative Medicine and the Shriners Hospitals for Children. And his research focuses on forebrain development in health and in neurodevelopmental disorders. I'm really looking forward to this data blitz. It's going to be a highlight of the day. So I'll turn it over to Kostas.
Thank you very much for the introduction, Kim. So for the first time, NeuroFest brings you a new and exciting competition called NeuroBlitz. NeuroBlitz challenges our graduate students to sum up their research for the general audience in a concise yet informative way, similar to an elevator pitch. Each student will have 3 minutes to present their research. As a member of the audience, we need your help. Please vote for your favorite NeuroBlitz presentation based on how well the students engaged with the audience and how clearly they communicated the significance and key concepts of their research. Once all of our contestants have given their brief talks, we will launch a poll on your screen to collect your vote, and the student with the most votes will win a $100 award. The winner of today's presenters will be announced during the interactive booth at approximately 5:30 p.m. And now I'm pleased to introduce today's NeuroBlitz graduate student contestants, starting with Lindsay Cameron, who is a fifth-year neuroscience graduate student studying the therapeutic effects of psychedelic compounds in the lab of Dr. David Olson. Please, Lindsay.
Almost everyone here knows someone that suffers from depression. It's a common and crippling disorder that affects about 1 in 5 people. Current medications are inadequate. So inadequate that suicide rates across the country have increased and now surpass the total number of deaths caused by car accidents. The CDC has referred to this as a mental health crisis. So why are these medications so bad? Well, there are 2 main reasons. The first is that it takes on average 6 weeks for these drugs to start working, leaving no therapeutic options for those that are severely depressed or suicidal. The second reason is that over 30% of patients are termed treatment resistant, meaning they don't respond to any of the medications available. Part of this problem stems from the fact that these drugs were invented decades before scientists really understood how these illnesses worked in the brain. But after these decades of research, we now have a much better understanding of how the brain works, and so we can develop better ways of treating it. And this brings me to talking about psychedelics. You've probably heard about them in the media, things like LSD or MDMA or magic mushrooms. They've been proven to work in human clinical trials to rapidly reduce depression within hours, and they are also effective in treatment-resistant populations. They work by strengthening parts of the brain responsible for achieving goals, motivation, and focus, and dampening down other parts of the brain responsible for fear or depressive thoughts. Now, as promising as these compounds might be, they're still hallucinogenic, and therefore they're dangerous. With the prevalence of depression across the country, we can't exactly have all these people going around tripping on LSD. So one question we can ask is, do you actually need hallucinations in order for this therapy to be effective? In other words, could you make a non-hallucinogenic version of a psychedelic? This is what I did for my PhD. We created a library of new psychedelic drugs. By doing minor tweaks to the chemical structure, we are able to create non-hallucinogenic versions. We tested them side by side, the hallucinogenic counterparts, and found they both increased the growth and function of important brain regions. They work rapidly, within 30 minutes after dosing in animal models of depression. In some tests, they even work better than the hallucinogenic drugs. I want to conclude by saying that we are on a path to developing a better, safer, new generation of neurotherapeutics, ones that work for everyone, so we can all be happy. Thank you.
Thank you very much, Lindsay. And this brings us now to our next speaker. So this is Stella Petkova, who studies in the lab of Dr. Jill Silverman at the UC Davis Mind Institute. She works primarily with mouse models of neurodevelopmental disorders, including models for Angelman syndrome and 15q duplication syndrome. Please, Stella.
Hi, everyone. I'd like to walk you through my morning. I woke up, walked over to the bathroom to brush my teeth, then to the closet to dress myself. I walked to the kitchen and made an omelet, and then walked to my office. Riveting, right? Exactly. No one thinks my morning is exciting, not even me, because I have normal motor abilities, so all of those things are easy and routine. For people with autism, any one of these could be incredible challenges. Today, I'm going to talk about walking with autism. Autism is a disorder that people associate with smart people who are quirky and socially inept, like the good doctor. But very few people diagnosed with autism end up being geniuses or able to perform surgery, yet over 80% will have motor skill delay and impairment. To study autism, we need to look beyond social behavior. So I look at walking. I study motor abilities in autism mouse models because if a mouse is going to help us understand autism, we need to look at the most impactful deficits. Like me this morning, most of us probably take our abilities to walk, bike, run, even write for granted. But for people with autism, even a few steps may require a lot of effort. This is a video of a typically developing 3-year-old walking on a pressure mat that tracks her footsteps. Like most of us, this comes with ease. Her footsteps are straight and narrow. She walks with a casual tempo and balance. This is her older sister, 5 years old, who does not walk with the same ease. She has autism, and like most kids with autism, she had delays and problems in walking independently and will struggle with coordination and balance for the rest of her life. So how do I mimic that with mice? I first track when mice learn to walk. Mice first crawl, then learn to walk slowly, just like babies. I watch how long it takes a baby mouse to walk out of a small circle. The autistic mice have difficulty walking and take longer to move out of the circle, indicating the same delays seen in humans. As adults, we observe their walking pattern on a mouse treadmill, which simulates the pressure mat the two sisters walked on and provides us the same information. I found that our autistic mice exhibit the same walking patterns as the autistic older sister, such as her wide, wobbly stance. These similarities validate our model and allow us to test therapies to alleviate motor disability. Maybe you think walking is simple and doesn't really matter, but consider what you did this morning, how your morning routine probably didn't include having to think about using your muscles, coordinating your limbs, and putting all your effort into just not falling over. One day, the same will be true for all kids with autism, and walking will be as mundane as my morning. Thank you.
Thank you very much, Stella. And this brings us now to our final speaker for today. And this is Alexa Dambra. She's a fourth-year PhD student in the lab of Dr. Diacino Fioravante, and she studies the cerebellum and how it influences motor circuits that are important for motivation, learning, and addiction. Please, Alexa.
You may have discovered at this point in NeuroFest that we have discovered quite a bit about the brain. And a lot of what we've learned so far can be described in diagrams like these. What these diagrams essentially tell us is that every part or region of the brain serves a particular function of our everyday lives. So the temporal lobe is for hearing, the occipital lobe is for seeing, and these diagrams tell us over and over again that the cerebellum is for coordination of our motor movements. So the cerebellum puts one foot in front of the other. It makes sure we get food to our mouth and not, well, everywhere else. Now, the way our brain cells or neurons communicate with one another in order to perform these functions is a lot like a game of telephone. You might remember from elementary school days, or maybe you've just seen it in the movies. But what you can do is talk into one cup. The vibrations of your voice travel all along the wire and are amplified by the second cup. So the person on the other side, no matter how far away they are, can hear what you said. Well, in the brain, every neuron has its own cup. And these cups have wires that can stretch to any other part of the brain. In my lab, we can shock the cerebellum neurons into talking, and their message is carried wherever their wires go. We know that the message is carried to certain brain regions that help transfer the information to the spine, because that's how we talk to our muscles and create body movement. We were surprised, however, to see that the cells in the nucleus accumbens had a second cup. They heard this cerebellar chit-chat, and they even changed their conversation based on what they were hearing. This is one of the favorite things about what I do. In my lab here at UC Davis, we discovered the first brain circuit that connects the cerebellum to the nucleus accumbens. Now, I said it was surprising that these two regions are talking to each other. Well, why is that? Well, in this same history of neuroscience that gives the cerebellum the one job, it also has a role for the nucleus accumbens. And this role is a reward center. So it's responsible for motivation, drive, interest, even some positive symptoms we've heard about like hallucination. Well, these have nothing to do with coordination. So is it possible the cerebellum has more than just one job? It wouldn't actually be all that surprising. Because fun fact about the cerebellum, while it's only 10% of the total mass of the brain, it contains about half of the total number of neurons. That's a lot of chatter for just ensuring you don't trip up the stairs. And so while we use this new information to shake up our old diagrams, we can also look to new roles for the cerebellum. So whether we're studying dysfunction that affects motivation and attention in young students, whether we're looking for treatments to drug addiction, or if we're just searching to learn about ourselves, why do I love broccoli while my dad despises it? We can look at the cerebellum for answers where we never thought to look before. Thank you.
Thank you very much, Alexa. And also to all of our other NeuroBlitz graduate students. This was an excellent and fantastic job. Now we'd like to invite you, our attendees, to vote for your favorite NeuroBlitz. I'd like to ask Teresa to please launch the NeuroBlitz poll, which you will see on your screen momentarily. Simply select the name and topic of the NeuroBlitz you enjoyed the most and submit. The candidate who receives the most votes will receive a $100 award, and I will look forward to announcing the winner today at maybe 5:30 p.m. So while you cast your vote, I'd like to return the screen to Dr. Kim McAllister. Thank you.
Okay, thanks so much, Kostas. And aren't our students absolutely amazing? They did an incredible job. And that's the tip of the iceberg. Wait till you see the interactive booths that they have. So, so that sure was a lot of fun. So I'm going to share just a few final remarks now, just for a couple of minutes. I want to make sure that you all know about our interactive booths. If you could go to the next slide. We have activities for all ages, from young kids to the young at heart. Could you go to the next slide, please? Great. So these are some of the interactive booths that that you'll be able to experience on the Gather platform. They're only available on the Gather platform, though. So if you could go to the next slide. The booths you can find them by clicking on this link. So Jennifer, if you could please add the access link into Gather within the chat, so those participants who have not already registered for Gather, or that don't have this link easily found, they can just go and click in the chat. So the interactive booths are going to be open for the next 2 hours. So let's go to the next slide. So if you did not sign up to visit the interactive booths, but you want to, I hope you will, there's no problem. You can go ahead and click on this link. You can log in using the email address that you used to register for this particular event. You're going to need to go through a one-time verification process, basically where they send you an email, check your email, respond to that. Super easy. I did it last night. And you can go to the chat to look for a step-by-step instructions here. So Jennifer, if you could please add the link into chat, that would be great. I think I saw you do that. For the attendees, if any of you guys have any trouble accessing Gather, we're going to have a couple of our staff members remain in the Zoom room for the next 30 minutes to assist. So you can simply use your Zoom link to re-enter this particular room if you've got questions. And if you enjoy the interactive booths today and you want to come back tomorrow, maybe with your kids or some friends, your Gather login will work for the interactive booth section of tomorrow's event as well. So finally, I want to again thank the people that made this happen. NeuroFest would not have been successful without the dedication and hard work of the NeuroFest event committee, staff, and volunteers. So first, I'd like to thank again the NeuroFest co-chairs, Drs. Tim Hanks and Kostas Sarbalis. We have our graduate student leads. They do so much of the work. Olga Vafaeva, Olga, I'm messing up your last name, I'm so sorry. Olga Vafaeva, Sasha Mikhailova, and Becca Wilson. Then I also want to thank our NeuroFest staff, Teresa Gear, Kimberly Cummings, and Faye Viers, and of course, Jennifer Scott for producing the whole event. And then again, all of our graduate student volunteers and faculty who were involved in the interactive booth experience, which really is the highlight of NeuroFest. So again, next slide. Thank you for joining us today. And if you've signed up to join us for tomorrow's session, I look forward to seeing you again soon. Thank you.